In this video, we're going to briefly learn about the 3 types of RNA because each type has a role to play in the process of translation. And there are 3 types of RNA. Let me get my pen going here. They are mRNA, rRNA, and tRNA. And each of these letters, the M stands for messenger. This is messenger RNA. The R stands for ribosomal or ribosomal RNA, and the T stands for transfer or transfer RNA. All right, now they're all forms of RNA, so they're all strands that have backbone made of ribose and phosphate groups, and then nucleotide bases sticking off, G's, A's, C's, and U's. But they have different functions and a somewhat different structure to each other. So messenger RNA, we have already learned about. It's the piece that's copied directly from the template, and it's the long, strandy one that has a cap on one end and a poly A tail on the other end, and it leaves the nucleus and it goes and gets translated. So that's our messenger RNA. The next couple slides, we're going to look at ribosomal RNA and tRNA and learn about them because they're going to play a role in translation when we get to that video. So first of all, we're going to focus on ribosomal RNA. So the reason this is called ribosomal RNA is that it makes up the ribosome. When we've looked at pictures of cells before, we've shown the ribosomes as little dots, little kind of filled in dots, right? But we've never really focused in on what are they actually made of. And what the ribosome is made of is a kind of conglomeration or a goop together of proteins and rRNA molecules. And so this picture here, the ribosome is shown in the brown, but I kind of squiggled over it and I drew these shapes here to show the proteins as solid lumps and the RNA as like squiggles. It's a really bad picture, I agree. But maybe a better way to think of a ribosome is a giant tangled glob of spaghetti and meatballs. The meatballs are the protein molecules and the spaghetti tangles are the ribosomal RNA molecules. And the whole thing kind of tangles up and kind of congeals together as a big lump, and that's why we draw them in cells as like little solid dots. But really inside that solid dot is a big tangle of RNA, ribosomal RNA and protein molecules, spaghetti and meatballs. Here's another picture showing the ribosome actually with the molecules in it. So if you look at this one down here, the teal and the gray are the ribosomal RNA molecules, and then the purples and the dark blues are the protein lumps. And the whole thing is kind of tangled up together to make this lump, right? So this is what a ribosome is made of. And this one here you can see spinning around. This is a ribosome from a bacteria called Deinococcus radiodurans. It's actually a bacteria that's incredibly resistant to radiation. That's why it's called radiodurans. It can endure radiation. But you can see here the green is the proteins, and the gray is one kind of ribosomal RNA, and the pale blue is another kind of ribosomal RNA. And it all tangles up. All right, well anyway, that's the ribosomal RNA. Not super exciting, but it's kind of good to see this diagram. The more interesting one is the tRNA, the transfer RNA, and these look really different. So tRNA molecules have this very distinctive shape. This is 3 different ways of showing the same thing. So diagram 1, diagram 2, diagram 3, they're all showing the same thing, just in different levels of detail. So to make a piece of transfer RNA, you have a long strand of RNA and then it kind of folds back on itself in what we call a hairpin or clover leaf. So this thing here, sometimes it's called a hairpin. That's like a bobby pin, you know, that you put in hair, a piece of metal that's folded back on itself, or a clover leaf, because you can see it's got three sort of bits that stick out like a clover leaf, shamrock or something. When the molecule makes this kind of folding back on itself, it creates this shape, and there's two parts that are important. One is the bottom down here that we call the anticodon, and one is the top up here, which we call the amino acid attachment site. I like to think of it as the head and the butt, all right? But it's not, that's not the scientific terms, right? The head is where an amino acid attaches, and the butt is actually called the anticodon. And the anticodon really is the three bases that are kind of dangling off the bottom here. This one's AAG. That's the anticodon. And this one has an amino acid attachment site, but there's no amino acid up here right now. It's just naked. All right? But it doesn't matter how you draw the diagram of the tRNA. As long as you show the head and the butt, or the anticodon and the amino acid attachment site, you're good. So this one is like a diagram showing the clover leaf structure. This one is showing more like how the strand actually bends around in real life. Here's the anticodon, here's the head, the amino acid attachment site. But honestly, those are all really hard to draw. So I like to draw these kind of candle shapes. Diagram 3 here. You've still got the most important parts highlighted, which is the amino acid attachment site at the top and the anticodon at the bottom, showing the three bases dangling off the bottom. So they kind of dangle down like this. They're not paired, like AAG, just kind of hanging down, and then there'll be an amino acid attachment site on the top here. So 3 diagrams of the same thing. The tRNA always has this kind of funky shape like this. All 3 RNAs, mRNA, tRNA, and rRNA, all work together to make the process of translation happen. And that's what we're going to talk about in the next video.